The recording before we had finished the last few slides, but I went ahead and made a new slide for you since I had mentioned don't confuse natural killer cells with cytotoxic T cells. They both release granzymes and porphyrin to kill their target, but they do this in a different way. Okay, so natural killer cells you saw on the first lecture, it's part of the innate immune system, so that means it's non-specific. Natural killer cells are going to kill any cell that are missing a self-tag. Okay. CD8 cytotoxic T cells are different. They need to be activated by a helper T cell, and then they're going to target the specific antigen that the helper T cell confirmed. And that's going to be displayed on the MHC1 tag. So instead of missing an MHC1 tag, these are MHC1 tags that have a foreign antigen on it or that have become strange, like in cancerous cells and even some infected cells. Okay, some cancerous cells and some infected cells will miss their MHC1 altogether and they'll be killed by natural killer cells. So only natural killer cells can kill the cells that are missing MHC1, and cytotoxic T cells can only target specific cells that they've been approved to target by the helper T cell. And so they're specific, they're part of the adaptive immune system, and they're part of the cell-mediated branch of the adaptive immune system. So hopefully that helped clarify it. So when I said make a compare and contrast chart, this is what I meant. Going back and gathering key information to help clarify and solidify information in your mind. You can do this for all sorts of stuff. It's a great study method. Okay, so next, HIV and AIDS. HIV is the virus. People can be infected with the virus and not have the disease. The disease is AIDS, which is takes a long time and is very, very awful. And the goal is that we're getting people tested and preventing the development of AIDS. So HIV stands for Human Immunodeficiency Virus. It is a super cool virus from the standpoint of a virologist in that it's unique. It's the only human virus that has the ability to go backwards from RNA to DNA. And so this is why we call it a retrovirus. It goes backwards. Its genetic material in the virus is already RNA, which then should just go to making protein and making new, making a whole bunch of new viruses very quickly. But instead, it goes backwards and it makes DNA. This is done by a special enzyme. Then there's another enzyme that allows it to integrate the DNA into the host cell that it infected. Acquired immunodeficiency syndrome is AIDS. Acquired immunodeficiency syndrome. And again, this takes time. We first declared this disease in 1981. HIV replicates in helper T cells, which are CD4. And this is primarily in the mucosa of the GI tract. T cell count should be around 1,200, but someone with AIDS, the virus has been replicating over a long enough period to destroy so many T cells that now their count is at or below 200. So you can imagine if your T cells have been knocked down to this low of a level, you're not going to have very much of an immune system, right? You need helper T cells in order to activate the B cells. You need helper T cells in order to activate cytotoxic T cells, right? So you're pretty much knocking out your whole adaptive immune system and have to rely only on innate. Okay, so HIV is an enveloped virus. 1 second, sorry, my cat is crying. Okay. And it has receptors on the envelope that bind to the host cell. And in this case, the host cell typically would be the CD4 helper T cell. Okay. And that allows the virus to be released into the cytoplasm. And remember, I said there's a special enzyme that allows it to take RNA, go backwards, and make DNA. That special enzyme is called reverse transcriptase. Reverse transcriptase only is found in retroviruses, and the only human retrovirus is HIV. Then it integrates this newly made DNA into the host DNA, and it does that with the second unique special enzyme called integrase. It's actually not written on this slide, but integration by integrase, ending with ASE for enzyme, right? So HIV has two special enzymes that allow it to go backwards and integrate into the host. Reverse transcriptase takes the RNA transcript and goes backwards and makes DNA. Nothing else, no other organism has the ability to do this. Okay, then integrase is another enzyme that allows it to integrate this newly made DNA into the host's DNA. And then as cells of the host cells replicate, they will replicate their own DNA with the viral DNA. It actually can't distinguish between it. And then that's going to lead to producing low levels of the virus. But this continues to happen over a long period of time. And eventually, this will happen to the majority of the T cells. Okay, tumor immunology. So we want to kill cancerous cells with our T cells and our natural killer cells. So natural killer cells if the MHC1 tag's missing, and killer T cells if the MHC tag has antigen on it. We have lots of chemicals and UV light, right? Different things that can cause damage to a cell's DNA and cause it to have uncontrolled cell division. So any chemical or any other substance that causes damage and leads to uncontrolled cell division is called a carcinogen. Cigarettes are carcinogens, all sorts of stuff. But we know that some carcinogens are much more potent than others. So just because almost everything can be labeled a carcinogen doesn't mean that you should just ignore that label. Instead, you should ask yourself, how potent of a carcinogen is it? How common does it cause problems, right? Is it known to cause cancer in a lot of people, or does it take a strange exposure, right? That kind of thing. So anyways, carcinogen, anything that's been shown to be able to cause uncontrolled cell division, and that's going to lead to the formation of a tumor. A benign tumor would be something that's very slow growing and it stays in place. It's not spreading around. A malignant tumor is able to grow very quickly and it will have the potential to spread. And so we say that it undergoes metastasis, and metastasis cells break free and spread and metastasize, right? Forming new tumors everywhere that they land, right? So treatment of cancer includes monoclonal antibodies. If you can make antibodies to specifically target the cancer, then you'll have phagocytes come and get rid of that cancer, right? So this is a very, very good treatment. We don't have this for all types of cancer. So you'll actually see all sorts of different treatments for different types of cancers, and usually a combination of treatments to make sure that you get it. When carcinogens damage cells, the specific area that it's damaging cells are the DNA, and again, more specifically, those are called oncogenes. So you've probably heard the term oncology, which are doctors that work with cancer patients, and so that's specifically dealing with the region of the DNA called oncogenes that have been altered in order to, you have to suppress the genes that prevent cancer, and then you have to turn on genes to allow uncontrolled division. So there's actually a lot of different genes that have to be modified. And so in a way, cancer is kind of difficult to occur, but we have so many cells that that it still actually happens quite often. And usually your body is able to get rid of it before it turns into a tumor. But if your body's not able to get rid of it, then it can become a tumor. And again, that could be a benign tumor or a malignant tumor. Malignant tumor is what you consider cancer, okay, that requires treatment. Immune system malfunctions, autoimmune diseases. So you can see your textbook for a list, but in short, we will look at hypersensitivity or allergies. The allergen is not actually a pathogen, but your body identifies it as a pathogen. Something like venom from a bee, mold particles, dust particles, dander, skin of animals. It could be food even. A lot of people have been hearing about gluten. For some people, very few, but some people actually have an actual allergic reaction to gluten. But a lot of people that say that they are not necessarily truly. You have to have a test done to know if you actually are. Other people, they might just have a little intolerance. It might upset their stomach, might cause bloating, etc. But an actual immune response is going to cause severe inflammation in the intestine, and that can have a lot more symptoms than just bloating and gas. Okay, well, bloating is gas. Okay, and then pollen, and of course, pretty much anything that you can think of could be an allergen. It's just something that the body has identified as a foreign antigen that it thinks is a pathogen. So an allergic reaction happens. This allergen or foreign antigen that is actually, it's not pathogen, but the body thinks it is. It reacts with the B cell, goes through the whole process, right? You have the helper T cell, and then you get a plasma cell that secretes antibodies. And the type of antibody that's being secreted are called IgE antibodies, and these are particularly found in your mucosa, so your nose, your eyes, your gut, right? And those IgE antibodies, in addition to some free-floating circulating IgE, the majority are going to be bound on mast cells. And mast cells, what their job is, whenever they get irritated or activated, they release histamine and other chemicals to cause inflammation. Okay. And you experience this inflammation from histamine as itchy, watery eyes, runny nose, maybe postnasal drip, sneezing, right? Okay, 2 types of allergies, immediate hypersensitivity, plasma cells that produce IgE antibodies. Antibodies signal mast cells and release histamine. Smooth muscle constriction of airways can occur. And if that happens, that's anaphylactic. It can lead to anaphylactic shock, and then you would need an EpiPen, right? Other people have the release of histamine, and it just causes normal allergies, like I talked about, sneezy, itchy, right? Eosinophils are a type of white blood cell, and they will then be attracted to the area because of the signals that the mast cells have released. And like I said, if you get too much of this, it can cause anaphylactic shock. Delayed hypersensitivity, you don't have this immediate itchy, watery, sneezy reaction of the upper respiratory system, but instead you have a delayed response, and this is poison oak, poison ivy. They both will trigger a delayed hypersensitivity. This is not the mast cells, not the eosinophils, not the IgE antibodies. Instead, this is causing a cell-mediated, so T cells, not the B cells, cell-mediated response. So then it's not even using histamine. So antihistamines are not going to help someone with poison oak or poison ivy. Poison oak symptoms appear in about 2 to 3 days, and you'd have to use a cortisone treatment to get the swelling and itching down. Okay, now we're actually at the end of the chapter. Email me if you have any questions. Bye.